Good evening, doctor. Welcome.
Hello, I'm Akira.
Hey, good evening, Akira. Nice to
Good
meet you.
evening. Nice to
Victoria.
meet you, Victoria. Yeah, nice to meet you. Yeah. I'm so happy to be here. Yeah, see you later. Yeah.
Yeah, we're happy to have you. And we don't really start until 6 o'clock. So if you want to just relax until then.
Okay.
Yeah.
That means 6 o'clock. No, 9
Yes.
p.m. 9 p.m.
Yeah.
Our
Oh,
time is
yeah.
now
Sorry.
10 p.m.
Yeah.
Yeah.
9
Yeah,
p.m.
okay.
EST.
Yeah,
Yeah, I'm on the West Coast.
yeah.
Yeah, so
Okay,
9 p.m.
okay.
EST, if you could, you could just mute your mic and hang out. That's great. And I'll see you soon.
Okay.
Thank you, Akira.
Yeah, see you soon. Yeah, thank you, Victoria.
All right. Okay, thank you.
Hi everyone, how are you?
Hello. Hi, Katarina, Jamie. Akira is here, and she's hanging out till we get started at 9 EST.
Hi everybody.
Akira, if you could make us moderators. So how you do that is you click on our profile picture, and then down on the bottom should be a button, make moderator. If you could, then I can share the link and things like that. Thank you. Akira, can you hear me?
I told Akira that we were going to start at 9 EST, and so he could relax until then. So.
Oh, but he needs to make me moderator first.
Yes, I neglected that little part of.
He cannot hear us.
Yeah. He might be away from his phone.
Okay, and can we close the room and open again? No, we can't.
Yeah, no, it'll be off the. It'll be off of the schedule. He'll probably be back. We can message him.
It's fine. It doesn't matter if the room is. I can just really quick recreate a new one. No. I'll try
We
to.
could, but it won't be, anybody who has the old link won't be able to use
Okay.
it. That's the thing. I just messaged him. Not that he's going to see the back channel, but, right, because we can't pin a link or a ping.
Okay. How is everyone? Good.
Good. I would imagine he'll be back. Yeah,
I,
there we go.
I got a message from you. I'm
Yes.
Akira.
Hi.
Yes,
So
yes,
first,
yes.
first a meeting. Hi,
Yes. Can
morning.
you
So
do us a favor so I can put the link up for your presentation and things like that to make us moderators? The
oh,
person that
how?
opens the room for it. So you click on my profile picture, and then there
Okay.
should be an option on the bottom
Okay,
saying,
make a control. Okay, now
"Thank you so
are
much."
you moderators? Okay, okay,
Yeah.
good, good.
Thank you. How are you
Okay,
today?
yeah, I'm fine. I'm fine. How are you today? How about
Good.
Katarina?
Good.
Yes, I
Thank
also
you.
good, good. Also, I sent you the file, so maybe you already receive it. So please
Yes.
use that for my presentation. Yeah.
Yeah,
And
I saw it.
I'll give you the request to click, just click and enter button, and that's enough. Yeah, maybe you can enjoy the some movies. Yeah,
Yeah, that will be
yep,
wonderful.
yep. Thank
I
you.
will put it up right now, and then everyone can check if it's working. 1
Okay,
second.
okay,
Okay, there. Please check everyone. I think it should work. The link to the presentation should be a really, really pretty presentation. Thank you. Thank you. I will check that, Rice. Thank you so much.
Katarina, I got an unauthorized when I clicked on the link. Is that the Google Drive link? It might just be
Yes,
me though.
yes, Rice. It should be. I have it on the right.
As long as everybody else's is working, it's fine. I just was
No,
informing
no,
that.
Rice said the same thing, but I don't understand because it should be working. So let me, because on my Google Drive it says it's free for everyone to view, but let me try again. 1 second. Oh, Akira is gone. Oh no. He left. It was for sure an accident that he pressed the wrong button. The file is really big. On my screen it says loading Google Slides.
Yeah, it says that on the room, so I think it's loading it. The pin is
Everyone
loading
try
as well.
again. And Akira's. Because on Google Drive it says it's public. but somehow it's not working here. So on my side it's working, but it says that it's... Oh, it works now? Okay, thank you, Rice. I really appreciate your help. Thank you, that's so nice. Okay, so it's working now. Now our guest speaker needs to come back. Let me share the link with him. Okay, let's see. I shared the link again. He should be back any minute. There you are. Perfect. Can you hear us well?
Yes.
Okay, great.
I I lost my my room.
Yeah, it's
Yeah.
easy sometimes
Yet,
to click the wrong button.
yeah.
It's fine.
Yes. I'm a totally beginner of this clubhouse. So yeah. I don't know. I couldn't understand what's happening.
Yeah, it's fine. It's happened
Okay.
to
Anyway.
all of us,
Anyway.
I think.
I shouldn't touch any button.
You're good. You're fine. Thank you
Yeah.
for coming and doing this adventure with us.
Yeah. Yeah.
Thank you.
Okay. Yeah.
We really appreciate it.
Yeah.
But in general, it's kind of fun. At least a lot of
Yeah.
people enjoy it, unless they are just very friendly. Then I don't know.
Yeah. Okay. I hope so. Yeah.
Yeah. So yeah, we are really excited about listening to this presentation. It's um,
Mhm.
it's very interesting.
Okay.
So
Thank you.
we'll start in around 2 minutes, everyone. Thank you
Okay.
for coming.
Yeah.
It's very nice to meet you, Doctor. I'm actually quite excited about this talk. Even the room title looks incredibly fascinating.
How's your day been, Jamie?
It's been great. Thank you very much. How's yours been, Victoria?
It's been lovely. I got locked out of the house accidentally, though, but I was able to climb back in a window when I found a really big ladder. Thanks to all my
That's
experience as a teenager breaking into the house at night after parents were asleep.
That's really lucky. That's always my worst fear. I always check my keys 3 times before I go out the door. I'm
Well,
so paranoid for that.
the wind blew the door shut. I didn't even know it was locked, and I wasn't. So it was a really
Oh
fun
no.
surprise. Yeah.
Yeah. Climbing in through your own window.
Right. Kind of a upstairs window too. Yeah, it was fun.
Oh dear. That's amazing you did that. Well done.
Thank you. Thank you. I was so happy.
That's great.
The only sad part was that I had a popsicle that I had just opened, and I had put it luckily on a plate in the house, but by the time I got back in, it took about 45 minutes to locate a ladder and a neighbor to hold the ladder, etc., and the popsicle was melted. That's
Oh,
the only
so
sad
the,
part.
the, the story had a tragic ending.
It did.
I'm glad you're back in your home, Victoria.
Thank you. Well, I'm late to my walk, so hopefully my signal will be good,
Okay.
because now I'm walking, but we're ready to go.
Yep, we're ready to go. So, welcome everyone to the Science Society, and of course, a special welcome to Dr. Akira Kagugu. And thank you so much for coming and making a Clubhouse account and going through all this struggle that you had to go through the first few minutes. But it's all sorted out now, everything should be working. So, we can go ahead and start. Because we are very excited to hear your talk. But before we start, let me introduce you a little bit to the audience. So Dr. Akira Kaguo, he is associate professor at the Department of Chemistry at Hokkaido University in Sapporo, Japan. And he did his bachelor of Fisheries Science Faculty of Fisheries in Hokkaido University, Japan. And then he did his Master's of Sciences at the Graduate School of Science, Hokkaido University, Japan. And then he did his PhD at the same university with the title of Gel Biomachines Constructed from Muscle Proteins. That must be one of the coolest titles of PhD theses ever. So I hope you won already a prize for that. And yeah, his research evolves around non-conventional materials with self-propelling ability through energy consumption, what is generally known as active matters. And he's interested in their mechanics and dynamics. And he's also particularly interested in dynamics, mechanics, and swarming of these active matters. And he has been recently exploring the responsiveness of these active matters and swarms, and how they react to various stimuli, which are the key to unravel the underlying mechanisms behind group behavior of living organisms. So yeah, he is aimed to develop advanced materials with intelligence, responsiveness, and adaptability for different applications. So the name of his research field is molecular robotics. And yeah, he received different awards and honors. The Science Award from the Society of Polymer Science, and from the Ministry of Education and Science and Technology in Japan for Japan's Prize for Young Scientist, where he studied ATP-fueled soft actuator constructed from biomolecular motors. And the most recent award was last year, where he received the HFSP award, and that is combined with a grant program for the title Structural Damage to Axons Resulting from Repetitive Mechanical Motion. So We are very honored having you here. And Victoria asks usually 1 or 2 questions in general, and then the stage is yours for the presentation. So welcome and thank you.
Okay, can I speak?
Yes, sure. And if it's
Yeah.
okay, Akira, may I ask you a few questions before we begin your talk?
Okay, please go ahead.
Thank you very much. So, what I would like to ask you is to give the talk a bit of a human side and to learn a bit about you before you begin your presentation. My question to you is, can you think back in your life to a time that you noticed that you felt a particular affinity for science? Some time that you felt that science was the thing that you really felt connected with, and perhaps this is in your childhood or some time that you choose, but that's my first question. Thank you.
Okay. A little bit difficult question to me, but so Victoria, so could you please make your question more concise? So you want to ask
Yes,
me
yes.
how can I say
Uh,
why?
Victoria, it's very noisy in your background.
Here,
Maybe.
Yeah,
I'm
yeah, yeah.
moving. Yes, I'm sorry. Here, I'm going to move. I hope this
Okay.
is better. Is this better?
Yep.
Yeah, a little
Okay,
bit better.
good.
Yes.
Okay, here's my question. When, Akira, when did you notice that you really love science?
Okay, that
When
kind of
in your
question.
life?
Yeah.
Thank you.
Okay, thank you very much.
Thank
Okay,
you.
first of all, Katarina, so thank you for your kind introduction, and so thank you for having me, your clubhouse. Okay, let me start by replying to Victoria's question. So, when do I notice I can be a scientist? Actually, in my whole research works, I encountered several how to say big discoveries. One discovery was actually I encountered was actually at the master course students. So I was actually working on the biomolecular motors as tiny micro actuators, and then I saw that those actuators are very cooperative moving. So I was totally fascinated with that kind of phenomena. So that was that time I decided to move on to this field, scientific field. Otherwise, maybe I am not so interested in this field, but that kind of founding was only the things that bring me to this field. Okay, is that enough?
Yeah,
Katerina?
thank you very much. And beyond that, your interest in that phenomena, can you take us along to how did you get here to this current research from this point to today?
Okay. Yeah, and after that, so I was really interested in how to. Okay, then maybe we have to start with my. By the way, so Katarina, so do you have my slide? If possible, I'd like to ask you to share my slide, or I don't know
Yeah. We have
how
the
to make my
slides.
slide.
Yeah, we have them available now. Thank you.
Okay. Okay, and so actually cooperative behavior is also found in nature, everywhere actually, and the squirrelfish and cells, every kind of creature can move in a cooperative way. So just started with this kind of phenomena observed in nature. I try to make a best effort to control the swarming, and that is also first step to make a tiny robot. That is also today's main topics. Yeah. Okay. Then how can I start? I don't know.
So you can please begin. Yes, you can begin and you can direct us through the slides, and then
Okay.
following your talk, we can have a question and answer when
Okay,
you're
then-
finished and when you're ready.
But can I share my slide with this
Yes.
device? Or Catalina, can you?
Oh,
Yeah, she will explain.
so
Okay.
yeah, this is not a screen share, so everyone in the audience is looking at the slides, but everyone so everyone has to scroll through them themselves. So it's
Okay.
really
Oh,
good
good.
if you say I'm
Okay.
switching to the next slide now.
Okay.
That's very helpful.
Okay,
Thank you.
then if so, okay, let me start. So first slide. Okay, as Katrina introduced me, I have been working on the such kind of active matter for a long time. And in the past few years, I have been totally interested in the active matters, which is even more dynamic in nature than some materials. So yeah, active matters belong to the group of the materials which can produce a motion by converting their own energies, for example, chemical energy or photo energy, etc. As a result, active matters can autonomously perform work without external support. This feature of such materials make them totally different from any other materials. So currently, I'm dreaming that my dream is to solve the existing some problem, energy problem, etc., or improve the quality of life through such a scientific science of the active matters. Okay, and today I'm going to talk about our tiny robot recently developed. So may I ask you to click the slide? The next slide you may find there's some swarming observed in nature. And actually this is also quite universal phenomena observed on the variety of the scale from the micro to the macro. Now you can see the swarming of the cytoskeleton in the cell, or ant colony, or schooling of a fish. And so far, a lot of effort have been made to understand the principle of this very fascinating collective behavior. And also now it's possible to reproduce those collective behavior in an artificial system, for example, by using the self-propelled colloid or biomolecular motors. Actually, I'm working on the biomolecular motor to reproduce this kind of phenomena. And by the way, the big question, why do they swarm? So do they swarm to just please our eyes? Of course, answer is no. So it's also a matter of the survival, quite important. And some attempts are also being made to understand their strategies. In this case, functions. So it is considered that by swarming, they are able to acquire the characteristics, for example, as listed here, parallelism, flexibility, or robustness. That can be actually obtained by the single individuals. So please go to the next slide, slide 3. And here I showed some functions emerged from the swarming. And these properties are not only so helpful for the efficient transportation. For example, now you can see the collaborative work by ant, and also prevention from the risks from the predators. That is also the quite important things for the survivors. And also efficient locomotion or information transfers, light bottom part, yes, and also the overcoming the obstacles, and also self-healing, etc., etc. And the idea of taking such function into the material world can be also another strong motivation. That is actually my standpoint. And a new research field is also emerging in the field of the robotics, looking at such function of the swarm. So it's known as a swarm robotics. So in the next slide, maybe you can find some examples. Slide number 4. Here I show the swarm robot invented so far. For example, as shown in the left-hand side robot, they are working in a group successfully, transport a very heavy brick. And also in the middle, swarm robot is sending some informations. In this case, character of K is on display. So this is also very, they are also very working. Yeah. But unfortunately, the size of the robots remain in centimeter orders, and their number is in the thousands. So development or developing large number of the small robot is the remaining challenge, and this is also most essential to enhance the scalability of such swarm robot. So, but how can we reach the submicron or even less than submicron, I mean micrometer scale region? Now you can see the my target written with the red colors, red text color. So it's probably impossible to achieve this with a traditional so-called top-down method. So maybe bottom-up approach is only the possible way to bring us to this range. So please go to the next slide. Slide number 5. So now I'd like to tell you the definition of the robot. So maybe there are some definitions though, but at least we can say that robots are the system which has actuators and processors and sensors as shown here. To develop our tiny swarm robot, we integrated photochromic molecules named azobenzene and also DNA-based molecular computers shown in the middle, and biological motors as shown here. In this case, kinesin and microtubules are used. And those tiny robots are synthesized with the help of the chemical engineering approach. Yes. And we have named such tiny system as a molecular robot, as Katarina introduced in the introduction part. So may I ask you to next slide? Let's move to next slide. Slide number 6. So this is a design of a system. DNA not only functions as a processors, but also provide attractive interaction between the agent that play actually important role in controlling the swarming behavior. And now we can also see the azobenzene-based photoswitch, so yellowish structure in the left part of my slide. Okay, and so please move to the next slide, number 7. And this slide shows a little bit of chemistry and how to prepare the molecular robot in a bottom-up way. And modification of azide by NHS Estel to the tertiary amine allowed to proceed the following reaction. That is a kappa-free reaction. And also by branching DNA in the DBCO, that is a dibenzoesclerooctane, written in the maybe somewhere in the right or bottom. We can easily conjugate the DNA with the microtubules. The microtubules act as a agent or molecular robot. Okay, and next slide become a bit more complicated, but let me explain. This is also a quite important part of our studies. And here, azobenzene, that is a molecular photoswitch, is introduced into the DNA strand. The amidite monomer is polymerized in a DNA synthesizer to obtain DNA with desired sequence. The sequence of DNA used in this study is shown here, upper right side, and they have a very complementary sequences. And also you can see the 6 and 8 azobenzene, the litunin Z. And I introduce every 3 residue to increase the response to the light. And then when exposed to the UV light at, for example, wavelengths of the 365 nanometers, these as a group isomerize to cis form. And the resultant steric hindrance blocks the hydrogen bond between DNAs. That is also important function of the photoswitching mechanism. And this results in other words, the reaction between DNA strands become inactive. And also this isomerization can be reversed to the original transform by irradiating with a visible light above the 480 nanometers. And this way, the melting temperature before and after UV and this irradiation can be easily controlled or modulate. And also the melting temperature can be also monitored by UV spectroscopies, as shown in the right side figures. Now you can see the change in the melting temperature, 73.7 or 38.9 at the transform or cis form. And now it's possible to change the melting temperature in a wavelength selective manner. So this is the explanation for the slide number 8. So please click and move to the next slide. So now let me show you how such system would form the swarm. Now what you can see here is the red and green filament that are conjugated to the DNA, but with the different sequences. And these filaments have not yet migrated close together, but when visible light is irradiated here, these filaments come closer and form the swarm. Maybe you can see the formation of the swarm in the next slide. So this is the outcomes. Now you can see the living things like swarming of our swarm robot. It looks really so animals, but actually those are the artificial products. And swarms look yellowish due to the mixture of the red and green filament. So I hope you can enjoy these movies. And if you click the slide, next you can see the dissociation of the swarm. And here we apply the UV light, and then just after the irradiation of UV light, it breaks away into the single filament. And this process can be repeated several times. So if you click the enter button, there you can see the reversibility of this swarming and also dissociation. And y-axis is denoted as the association ratio, and then y-axis is a cycle I made it. 1 cycle, 2 cycle, 3 cycle, and maybe we can also repeat several cycles. Okay, and please go to the next slide, 12. And in this slide, I'd like to explain the controlling the mode of the swarming behavior that can be modulated by changing the stiffness of the filament. And the rigidity can be varied by a factor of 2 by using those components, DMCPP or GPP. Just you can mix those components, we can easily regulate the stiffness, or you can say rigidity of the the filament. And if we use the filament with a little bit flexible rigidities, we can get this kind of ring-shaped swarm. And this is so slide number 13. And if you click the button, you can now see the dissociation, the ring and shaped swarm, and that's a UV light. And now we can also see that this swarm is composed about 300 filaments. So in this way, now we can possible to control not only the formation of the swarm, but also the mode of the swarm behavior. Okay, then you saw what is the next. If we want to call this system robot, we have to let them do our work. So it is obvious if you refer the term of the robot. The term actually robot, the name of the robot comes from the Czech word robota. Robota that means the labors. So now we choose the cargo transportation as the most simplest task, as shown in the this video, slide. Now I'm transporting some cereals to some destinations. But I found that it was a not good choice, since next slide, to work as a transporter, at least 3 functions are required, or 3 functions should be considered, such as pick up cargoes, and transport the cargoes, and deliver to the designated place. So you can easily understand this is not so easy task. But at least we just launched this research work. We have no choice to proceed. And please click the button, and then now you can see the cargoes. So microplastic was used as the cargoes, and also to recognize the cargo, DNA tag was tethered onto the surface of the microplastics. Okay, and then if you click the enter button, then you can see how transporter pick up and transporter cargoes. Now you can see cargoes, green spot that is also cargoes, and reddish color is also our transporter, but it is single transporters. Then if you click again, now you can see that the green cargo was transported by the single transporters. On the another front, the group of the transporters are also able to carry the multiple cargo. So please click the slide, and then now you can see the cargo transportation by the swarm transporters. And now you can see the yellowish assembled is transporting the multiple cargos. Already we can see the many swarm transporters are carrying the cargo. And by the way, what is the advantage of such a cooperative transportation by a group of the transporters? So please click, then now you can see the trajectory. Maybe this trajectory provides you the very clear answer to those questions. So a group of transporters are carrying the cargo for a long distance. So please go to the next slide 17. And this slide also shows this slide shows the time dependence of the distance of the bees transported. Clearly, we can see that transport by the swarm is carrying the bees a long distance. Yes, and so that next, for all the bees, we sum up the distance traveled by unit time. So in this case, 5 minutes. So if you go to the slide number 18, now you can see the result. So actually, I was so surprised by this result. Now you can see difference in the transportation efficiency by the single filament, single transport or the swarm transporters. If you click, you can also see the differences, almost 10 times differences in the cell transportation efficiency. And I have been doing study for many years. This result was either the first or second most striking one. Maybe this is also most important part of the animals or the living things to form the swarm. And if you click, please move to the next. Slide number 19. This slide shows the transporter carrying the cargo with the different diameters. And now we can also see that beside the efficiency of the transportation, we also found that there is also significant difference in the size of the transportable cargo. The cargo size was in this shown here, that varied from roughly 1 micrometer to 30 micrometers. So movie on the top show the cargo transportation by the single, and bottom is by the group of the transporters. And single transporter could transport cargo up to just 3 micrometer in size. But in case of the group, cargo size as large as 30 micrometer could be transported, which is almost 10 times also increase the size compared to single transporters. So that's why the large cargos are only transported by the swarm, and those are also not transportable. Okay, and I'd like to move to the next slide. So let me summarize so far. That's way. So pickup and transport cargos were successfully demonstrated, but they are just actually scattering the bees from here to there. So they are not working from the thermodynamic viewpoint. So that next step is to deliver the cargo at the designated places. But how can I do that? So actually our transporter can form swarm under visible light, whereas UV light can trigger the dissociation of the swarm. So dissociation of the swarm may allow us to unload the cargo from the transporter. That is also idea. It's also a very simple idea. Then, so if you click the bottom to move to the next slide, now you can see the demonstration of the dissociation of the swarm at the designated place. Now you can see the visible lesion and UV lesion. Then as they get into the UV lesion, we can see that swarm gradually get smaller. Yes. And also it just, for me, I don't know, but they are trying to avoid this region. It looks like avoiding to enter in this region. Okay, then, so please click the slide. Then this is also a demonstration of the accumulation of the cargo. Now, some bees were introduced in this system. And the area surrounded the purple line, a little bit difficult to see, but in the middle is a place where we have UV light irradiations. And I guess number of the particle at the place was found to increase with time. So maybe from the movie you can see that. And if you move on to the next slide, 23, we can see the change in the density of the cargo over the time at the UV lesion, 0 minute or 60 minutes. Now you can see the change in the drastic change in the density of the cargos. And Also, this is an important thing. So I want to also remind you to focus on the visible region. So visible region mean the region we have no UV light. And if you compare the 0 minute and 60 minute, we can see almost no changes. That means cargoes are only transported at the designated place. Okay, this is a result, and also this result quite reminded me of the backtoss game I experienced at an athletic event when I was a child. So it's a game where you throw the ball into the basket on the high ball, and now you can also mimic this system. Now pole is the place where we have a UV irradiated region. Okay, maybe almost all of my talk. Let me summarize my talk with this slide. Our system is composed approximately 1 million agents with a size of 10 micrometers. And this tiny swarm robot could have a wide variety of applications that cannot be achieved by the mechanical machine or single entities. Okay, and this is also acknowledgement. Especially I'd like to acknowledge all the collaborator, slide number 26. And also, if you click this one more time, then you can see my face. And so the Dr. Mushimi, she's also first author of the paper recently published in the Science Robotics. Okay, maybe 30 minutes almost has passed. So thank you for your kind attention. That's all. Yeah.
Thank you so much for this
By the way,
presentation. Oh, yeah, go ahead.
okay. Mm-hmm.
Oh, I didn't want to interrupt you. Did you want to say something?
Oh yeah, and I just wanted to confirm that slides are so perfectly in monitors or not. Slide was okay.
Yeah, it was okay in the beginning.
It works. Mm-hmm.
Yeah, it did.
Okay, good.
Yeah.
Mm-hmm.
If I think for at least for most people it worked, I think. And then I got Serena the slide pictures because she's traveling, so our internet is not efficient enough to download
Okay.
to see the videos. But we managed to
Okay.
set.
Okay. Mm-hmm.
Thank you.
Yeah.
Serena, did you have a question? I saw you on my.
Well, yeah. So I didn't get to see the videos, but I
Oh, really?
was able
Oh.
to see the slides. Yeah.
Oh,
So I
my
do have
God.
a few
Yeah.
questions. I know. I'll be able to see them. I'm on travel and at a hotel, and I
I see.
can't
I
download
see.
them.
Yeah. But maybe
But
without any movie, maybe I cannot how to say explain
right.
how
Well,
fantastic.
so
So.
what I gathered is
Yeah.
okay. So you've got microtubules as the
Yeah.
transport. You've
Yeah.
derivatized them with a particular units
Place.
that go
Mm.
through with cis-trans isomerization with UV and DIS. Is that right?
Yeah. Yeah. Yeah.
And
Yeah.
they
Exactly.
aggregate. Okay, so then the microtubules can then aggregate
Mm-hmm.
and transport collectively, right?
Yeah. Yeah.
Okay.
Exactly. Yeah.
Now, and the cargo is DNA.
No, no,
What was the
cargo was
cargo?
the cargo the microplastics, polystyrene-based plastics.
Oh,
Yeah,
okay.
yeah. The cargo is not so DNA. DNA is just tethered on the surface of the plastic, microplastic,
I
to
see.
just identify the cargo. Yeah.
Okay. So
Yeah.
then, so the microtubules then,
Mm-hmm.
with the side chains in
Mm-hmm.
with the UV or vis, they can aggregate or disaggregate, and
Yeah.
so they're picking up the plastic.
Mm-hmm.
I'm unclear what is the the transport direction. Is there a gradient, or what drives them in a particular direction?
Oh yes, actually we didn't control any direction, but just we let it UV at the designated place. And then once they come into that place, they started to dissociate. Then again, associate after getting out of the UV lesion, since visible light is already available in the outside of the UV irradiated lesion, so they start to form the swarm. And then at the same time, they started to collect the bees around the swarm. And then again, it's just by chance, but again, if they found the UV lesion, they also release the bees. That's way we can collect the bees. That's
Right,
yeah,
right, right. Okay,
yeah,
so
yeah,
fascinating. That's really
yeah.
fascinating. So
Mhm.
wherever you point a UV laser is where the drop-off point's going to be, right?
Where? Yeah, yes, exactly. Yeah, then just making the photo mask, and then
Uh-huh.
or photo mask, or you can also directly irradiate the laser, UV laser by squeezing the focal point. Yeah,
Oh, that's
even
fascinating.
even you can make any kind of shape. Yeah, triangle or square, circular, any kind of yes.
So I have
Yeah,
a question that on the microtubules,
yeah.
and there's a lot of interest in that, but that we had a room earlier
Mm-hmm.
that looked at in the case where there's resonance,
Mm-hmm.
dipolar oscillations, and
Oh
between
yeah.
two molecules that have
Mm-hmm.
similar frequency, you get 1/r³ as opposed to 1/r⁶. I'm wondering
Mm-hmm.
if with the UV, if it's driving a more efficient aggregation because of these
Oh.
similar resonances.
Okay, that
Any?
good question. That is a maybe good question. And also there are some report. The microtubules are very reactive to the such kind of light, and then show some resonance. But at least my research work, I haven't considered anything about that kind of. So
Hmm.
yeah, let me try to yes. Yeah, thank you for your good suggestion. Yeah, yeah, that is a good point, I guess. Yeah.
I mean, because that
Mm,
would
yeah,
be an interesting effect to capitalize on, but
yeah,
it
yeah,
but it may
yeah.
already be operative here. So that's
Mm,
fascinating.
mm-hmm. Yeah,
Excellent
thank you
work.
for
Thank you.
yeah, thank you very much. Yeah.
Um, Thank you so much. I have a question about the DNA strands. How do
Okay.
you design them?
Okay.
How do you
Okay.
design them? Do you calculate like at what temperature they
Ex-
like? Do you yeah? And then, does
yeah.
pH level also influence your DNA strands?
Yeah.
How you design them?
Okay, good. So actually, we systematically change the DNA sequences by seeing the delta G. That is also important, and also the melting temperatures. So I, I pick up the DNA sequence with 609. That is the DNA which has a melting temperature very close to the room temperatures. So that is the yeah idea we design. Yeah, and also we also design DNA sequences with three repeated units. So to make how to say not robustness. How to say to make the duplex to form the duplex more easily. Yeah, that is also kind of idea. Yeah, is that okay?
Yeah, thank you.
Mm-hmm.
Yeah,
Mm-hmm.
and there are also ways of maybe using these loops. So pH gradients could
PH,
also
yeah, yeah.
influence the swarming behavior.
Mm-hmm.
Is that also something you may be working on?
Yeah, maybe. Yeah, but at least we fix the pH at the very neutral. So to work to how to say let our microchips work. Yeah.
Yeah,
Mm.
interesting.
Yeah.
Yeah,
Yeah. Yeah. Mm.
we had the room at some point where they use these gels with DNA and gold particles, and they use eye motifs to
Oh
make
yes.
um, Yeah, to make the
Yeah.
different colors with different pH levels. They
Yeah,
were.
exactly, yes. Yeah, that is also interesting. Yeah, exciting
Mhm.
part of the work, I guess. Yeah.
Thank you. Eli, did you have a question? I saw you on mic.
Yeah, thanks for the presentation.
Yeah, you're welcome.
I've worked with a number of related things over the years. I
Mm-hmm.
replicated Yanagita's groups,
Oh,
uh,
Yanagida,
um,
yeah.
the
Mm-hmm.
total internal reflection, fluorescence microscopy of
Mm-hmm.
actin-myosin. So
Okay.
just wondering, your choice of microtubules, was that because of the greater persistence length of microtubules?
Mm-hmm. So sorry, persistent? I cannot so catch the
The
last.
fact that they're more rigid, longer persistence length.
Yeah. Also reason, yes, it's microtube that's also long and also very rigid, so filament. So that is also, yes, main part of this work. Yeah. Is
So
that
yeah,
okay?
really interesting. Thank you.
Yeah.
Did you
Um,
stabilize the? Oh, I'm sorry.
no,
Go
don't
ahead,
worry.
Katarina.
Go ahead.
Did you stabilize the ends of the microtubule? I understand there's, or I mean, were they or dynamic in their length over the course
Oh,
of?
yeah. Stabilized with the so taxol or yeah, some chemicals. Yeah, already stabilized. But also there are some chance to also make their property more dynamic, and then in that case maybe we can see more different type of the swarming. Yeah, but I haven't tried that yet. But I'm interested in that. Yeah. Okay.
Yes. I wanted to ask. Oh, did I interrupt something? I'm sorry.
Well, I was going to ask a
Go ahead.
different question. So what was the most troublesome thing that came up as you were doing this work?
Oh, that is also a good question. So now what we have to use is the, so a little bit low wavelength of the light, and sometimes that causes also a negative side effect on the so protein. So yeah, for example, degradation or photobleaching or something. So that was a little bit so tricky part of my research work. And but to remove those kind of difficulties, we perform all the so experiment in the so quite inert atmosphere. So yeah, that was a most difficult part of my work. Yeah.
Dr. Shah, did you have a question?
Yes, so thank you so much, Akira. My question from
Mm-hmm.
you is about the, I mean, azobenzene that you just used
Okay.
that, and we know that it's a metal-free ferroelectric. So I
Yeah.
just noticed that there was differences in proportion of the trans versus cis, because we know that about the different photoisomerization behavior
Okay.
between the
Mm-hmm.
trans and cis. And I was just wondering, did you predict it on this? I mean, because I see the trans form more prominently rather than cis, or if just based upon whatever DNA that you have.
Actually, yes. So azobenzene, of course, azobenzene is a quite so how to say classic component chemicals, but we didn't know that. So this kind of idea really work or not. And actually, so if we have the so tertiary butyl, okay, this is a little bit chemical, too much chemical, but tertiary butyl group at the so end of the so as a benzene, we can have the so yeah, surprisingly we can have a totally opposite behavior. So now we have as a benzene, trans state as a benzene, trans state as a benzene, then enhance the interaction between the DNA interaction, and cis state is the opposite. But if we have some bulky compound at the end of the azobenzene, you can have a totally opposite. If you apply the UV light, we can have a more enhanced interaction between the DNA. So yes, this is just a kind of try and error experiment. So yeah, is it okay?
Yes, sure, thank you.
Yeah, thank you so much. I had another question. So
Mhm.
if you use this for drug delivery, for example, and ideally with precise, are you working or planning to work on a process to make the delivery like precise to a specific organ or specific cell types like cancer cells? Is, is there a way to you have built in basically an address that you could put them in?
Mm, a little bit so drag, your talk was dragging, but so your question was so application, for example, is it possible to transport some drug to the so some specific cell? Is that?
Yes, exactly. Let's say cancer
Oh,
cell
okay, okay.
or maybe
Ah,
you
yes,
know.
yeah. Yeah, this is just also preliminary research work. Well, now we are trying to not as a cancer cell, but just a living cell. For that targeting a living cell, we are trying to transport some, not a drug at this moment. Just transport some dyes or some fluorescence dyes into the into the cell. So now we are trying. So, but maybe yeah, we need to check the feasibility in the more elaborately, but that is also our dream. So yes, that question is quite reasonable. Yeah.
That will be so interesting. So so how so would you build into the DNA a recognition mechanism
Mm-hmm.
or surface like yeah, is there so basically could you build for the microtubules that they will recognize like a receptor of some signal? Is there
Yeah.
a way to
Mm-hmm. Okay, then so yeah, question is maybe asking about recognition of the not the DNA but so any other chemicals. So yeah, so DNA now they can be used as a kind of aftermath. So any kind more than 500, I don't remember, the 500 species are already reported to be recognized by the DNA. So that yeah, that means so we can also how to say expand the function of the our molecular robot system. I guess or I hope so. Yeah. Yeah.
yeah, that would be very exciting.
Mhm.
Did anyone else have a question? Jamie, did you have a question? Um, yep.
I guess I'm curious if if we could start thinking about multi-stage systems. So
Mhm.
if the original system can in essence collect and scavenge and drop off in a point where there's UV light,
Mhm.
that would effectively concentrate the cargo in a sense.
Yeah. Mhm.
If that could then if where it got concentrated was a second stage, for
Mhm.
example, that had better, more directional or directive transport properties, or it'd be interesting to have a cascade of stages where you could scavenge these things from the environment and collect them through
Mhm.
these different stages and transport them over much larger distances.
Okay. Yeah, thank you very much for your comment. Yes, so like molecular factories, yes. So once transport a cargo, and then that cargo can be react to the cargo transported forward. Then we can maybe yes assemble the some very specific structure, or you can also make some specific reaction to produce some special chemicals. Yeah, and to me for such kind of direction, maybe we have to at least incorporate or recruit the DNA which has the responsiveness in the multiple light. So that is actually ongoing topics. Yeah. So does my answer make sense?
It does, and yeah, it was essentially a comment about possible future directions. I guess the question would be, what's next for you in this work?
Oh, what next? Yeah. The next figure is as Katarina asked me. So transported some drug to the target cells, or we are now trying to also enrich our system to respond to any kind of signals, not only the light signal but the chemical signal, pH signal, yeah, etc. Yeah, that is also our current notion. Yeah.
Hello, Doctor. This was an
Hi.
incredible talk. Thank you so
Thank you.
much.
Thank you very much.
I was wondering,
Mm-hmm.
would this swarm thing ever be possible for the swarm to unite to become some larger thing, to like pick up something larger, then break apart again, like connect together? Is that the kind of thing that would ever be possible?
Mm-hmm. Mm-hmm. Yeah, at this moment I can also give you the complete answer, but maybe in the future. So, your question is you want to ask me the universality or something, yes? And
Yeah,
also
yeah, just
or
just
scalability, yeah,
universally, like for
scalability or something, yeah. Maybe in the future, yeah. Yeah, we can achieve that. Yeah, I guess.
that would be really
Yeah,
really
this moment.
cool. That would be really cool. Thank
But yeah, yeah,
thank you
maybe,
very much.
yeah.
Maybe we can have them build buildings by themselves.
Yeah.
We had a room where we discussed more like blue sky
Mm-hmm.
type
Yeah,
of ideas where people are trying to build intelligent buildings, and
yeah.
maybe one
Yeah,
day your swarms will build
yeah,
intelligent buildings better than we do.
yeah. But of course, there are so long distance to that, so please, but maybe.
Yeah.
Yeah. Also, that is my dream. Yeah. Yeah.
And there was a moat with AI alligators in it to protect the building.
Mm-hmm.
Oh
Mm-hmm.
yeah, our
So
buildings had immune system, so it's the safest security system. So if somebody comes that doesn't belong there, the building immune system would throw them out.
Yeah. Yeah.
Anyways,
Mm,
that was
yeah.
just more kind of a
Mm-hmm.
joke we made after.
Yeah.
We dream big here in Science Society.
Yeah.
Is there a oh sorry, I was going to ask, is there a limit to how big you can make the swarm before there's any
How
problems
how how
communicating?
how you make a big sorry,
You know,
how?
the swarm you're talking about, yeah.
Yeah.
Um, is there a limit to how large you could
Oh,
make the
large.
swarm before there's any
Yeah.
communication problems?
Yes, so yeah, that is a good idea. Good question. So if we have lot of agent, so you can increase the size of the swarm. Yeah, but at the same time, so with the size larger, the the viscous, okay, yeah, this is a little bit tricky or scientific, but yes, maybe there are some limitation. If you want to make them so active, so they started to also fight against viscous drug. So yeah, but so far I have seen the swarm with maybe sub-millimeter or something. Yeah. But the make them more bigger is through some how to say there are some need to or find some way to reach that place. Yeah,
Ah,
size is still yes, yeah, several hundred or something. Yeah, or less than millimeter.
okay.
Yeah,
Thank you very much.
yeah.
I have a question, please.
Okay, yeah.
I'm asking on behalf of Victoria in the audience. And her question is, what is the worst case scenario you'd imagine if this were no longer experimental?
No longer, you mean that's no, no.
Good question, it sounds like.
Yeah, yeah. You mean no longer experimental mean that, so what can I answer to that?
Oh, is this the anti-entropy apocalypse?
Yeah.
Yeah, I don't know right now if
Okay, I don't know.
yeah, because they are controlled by light and UV light, so they
Yeah.
are highly controllable. So there shouldn't
Yeah.
be any risk of them going wild.
Risk, yes. Yeah. Yes, that is a good question. At least, yeah, ethnical the viewpoint. So yes, we are using DNA. So DNA is quite interactive with the our system, and also now we are also working on the sensing the RNA, not only the DNA but RNA or something. So yeah, if you want to use our system in our body, maybe we have to more be careful about the usage of this system. So maybe yeah, that remind me of some kind of scientific fictions, right? Yeah.
So
Yeah.
so
Oh, fantastic voyage. Yeah.
Yes, exactly.
Yeah.
Well,
I
she added malicious. She clarified, not even going wild, but a malicious use.
even to use maliciously, I'll emphasize that this is with purified proteins
Yeah.
and specifically designed sequences of modified DNA. That I mean, even the DNA modifications are not something that would be very very easy to get your hands on, and all of this, as was mentioned, the UV light to not have it even damage the things that you're trying to use. You have to have this completely under an inert atmosphere, so no oxygen around. And if just like a bacterium got into this, it would just eat everything, right?
Yeah.
So this is highly controlled circumstances that are not easy to achieve, right? I mean, if somebody wanted to misuse this, I mean, you could misuse UV lasers themselves much more easily.
Yeah, great.
Yeah. So please do not
Could
misuse.
just use virus, right? Over here.
Yeah.
Modify it slightly if you want. Like this would be way too complicated to
Yeah,
so
I haven't thought about that. Yeah, but yes.
yeah, it's an hour now. So I hear that you have also work to do still because your day is just starting. So thank you so much for coming and sharing this wonderful presentation with us. And we would
Thank
be
you.
very curious to hear when you have news updates if it worked to deliver this into cells like the fluorescence. So
Yeah.
feel welcome to present updates at some point
Okay.
when you have.
Okay. At some
And
point.
I hope you
Okay.
enjoyed it. I know you were a little bit, not too comfortable to come to Clubhouse, but I hope you enjoyed the experience. And we, for sure, enjoyed it.
Yeah.
We had a lot of fun.
It was
Yeah,
a
thank you
great
very much,
experience.
Doctor. Thank you.
Okay, I will.
Fascinating work.
Yes, I will
Thank you so
update.
much.
So my research work at some point. Yeah.
Yeah, wonderful. Yeah, I really hope you enjoyed it. That was, we for sure
Okay.
did. And yeah, always come back whenever you have time and have
Okay.
something to
Okay.
share.
Yeah.
And we hope that you make a lot of progress. And
Mhm.
get a lot of funding and
Mhm.
and a lot of great students and postdocs. So yeah, we wish
Yeah.
you all the best for your research and
Yes.
thank you.
Thank you. Mm-hmm.
Yeah, and thank you everyone for coming and for asking great questions, for interacting with us. And feel free to join the club if you like discussions like this. We'll have our next room. Actually, we have a bunch of rooms this week. So we have on Wednesday, 1:30 p.m., Dr. Arshamian talking about his research that perception of odor pleasantness across cultures is actually has quite a lot of overlap. It's a really interesting cross-cultural research. And then in the evening at 9 p.m. EST, Dr. England will be here talking about Atlantic Ocean currents and how they are slowing down. And then on Thursday, Dr. Bennett, he will talk about a new treatment target for chronic pain. And Dr. Congreve about 3D printing with light converting nanoparticles. And on Friday, we'll have Dr. Santos. We have a lot of molecular machines. So she was a student in James Tour's lab, and she published a paper and finished her PhD. And she will talk about light activated antibacterial molecular machines. They have like little drills and damage antibiotic resistant bacteria and other microbes. So it will be also a really interesting room. Maybe Kira, you would like to join us for that one if you have time.
Okay.
But
Okay. Thank you. Yeah.
for you Saturday, so I'm not sure if you want to come on Saturday, but it's
Okay.
also about molecular
Yeah.
machines.
Yeah. 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 Yeah.
But if the
Okay.
timing's bad, the replay is saved, so you can always listen
Okay.
to it later.
The next show will be started at the same time, 9 EST.
So that room on molecular machines on Friday is on one.
Fr-
Is at so here? So in New York, it's Friday at 1 p.m. EST. So is
mm-hmm.
it 12 hours difference right now, or how many?
Mm-hmm.
Or
1
13
p.m.
hours?
for 14
Oh,
hours.
it's very late, right? 14
Okay.
hours. It
Mm-hmm.
probably is it in the middle of the night?
Okay.
I don't know. I
Okay.
can send you an email with the link,
Okay. Okay.
and then you can. It should be in your time zone, or
Uh-huh.
if you click
Okay.
on it, you should see it in your time zone. If not,
Okay.
we'll have
So
to report. Okay.
yeah, please send
'Cause
the link. Yeah. Yeah. Thank you.
yeah, she's in Europe. I think Europe and Japan don't have a lot of good overlap time.
Mm-hmm. Mm-hmm.
I think as well.
Okay.
Okay, thank you so much, everyone, and enjoy the rest of your day. Thank you for coming, and yeah, I hope to hear you all soon again. Bye, everyone.
Bye, everyone.